Hello, everyone, and welcome to the webinar. I'm Kate Osterly for Genomenon, and I'll be your host today. Thank you so much for joining us. Today we have attendees from all over the world tuning in. So wherever you are, we hope you are safe and healthy, and thank you again for being here. We have a lot of great information to share, so I'll get right to housekeeping and introductions. As you are watching, please feel free to submit questions through the drop-down window, and we'll answer those right after the presentation. Today, Genomenon co-founder and Chief Science Officer Dr. Mark Kiel will be presenting Big Data for Rare Disease, using genomic landscapes to accelerate drug development. Mark completed his MD, PhD, and Molecular Genetic Pathology Fellowship at the University of Michigan, where his research focused on stem cell biology, genomic profiling of hematopoietic malignancies, and clinical bioinformatics. As I mentioned, he is the founder and CSO of Genomenon, where he supervises the scientific direction of the Mastermind suite of software tools. Mark, ready to get started?
I am, Kate. Can I be heard?
Yes, I can hear you.
Okay, great. I'm going to take that to mean that the attendees can hear as well. Welcome, and thank you all for joining. I'll echo what Kate said. I hope that everybody is staying safe and healthy. I heard in a couple of recent meetings that people are getting Zoom fatigue, which I would have thought was an oxymoron, but is apparently a real thing. So all the more grateful I am that you guys have deigned to join us. As Kate suggested, I'm going to be talking about big data for rare disease and how that data can inform drug discovery, and specifically address how Genomenon can help in that endeavor. So this is the outline of my talk. I like to keep things simple, so I'm going to talk about why genomics can be used to great effect in rare disease, what are the benefits and specific applications, how you should go about it with specific reference to Mastermind Genomic Landscapes, which I'll clarify in some detail. And then the bulk of the conversation here, I would like to focus on some specific examples. So what are those representative case studies that Genomenon is at liberty to discuss? Before I begin, I like to keep things at a high level to start with, and to emphasize that Genomenon as a company has a core vision, a mission that was true when I founded the company and continues to be true for every one of our team members and in every one of our activities, and that's to drive rare disease patient diagnosis and treatment. Our goal is to have every one of these patients be appropriately diagnosed and receive the standard of care. And that's a real challenge when we're talking about rare disease, given some of the limitations of finding that evidence in the medical literature and surfacing that at point of care into clinicians' hands. So let's start the conversation here in the webinar with the why. Why genomics? Why particularly for rare disease? So this may be news to some of you who are attending from the rare disease realm specifically, but genetically supported targets double the success rate of drug development and delivery into the clinic. So if there's genetic data supporting your target, the biomarkers that your drug is actually targeting, that leads to a dramatic increase in the likelihood that your drug development program will be successful. And Genomenon specializes in identifying, surfacing, refining, and delivering that genetic data. More specifically, where there's great promise in genomics for drug discovery, as you'll see, one of the common threads here is this impacts a number of different rare diseases, specifically complex or heterogeneous diseases, where there's a need to identify either gain or loss of function genetic variants, whether there's multiple related disease types that can then be converged into a unified treatment strategy. Genetics and genomics and all of that associated data can drive that unification, which obviously has benefit for clinical trial design and development, as well as post-commercialization activities. Genomics can also resolve pathway heterogeneity in otherwise clinically heterogeneous diseases, or on the other side of that equation, can also uncover latent homogeneity that wasn't known absent any of this genomic data. And so depending on the starting point, genomics can lend clarity to those types of genetic conditions. And then the undercurrent of what I'm going to be talking about today is particularly true of rare disease, where there may be a perceived dearth of information. Identifying that information and assessing it in its completeness is one of the key value propositions of Genomenon's genomic landscapes that I'll be discussing in specific use cases here in a moment. So genomics empowers pharma to affect positive outcomes at all aspects of the drug discovery pipeline, from optimizing identification of preclinical targets to expediting downstream R&D activities, thereby reducing overall R&D costs, to maximizing the likely success of clinical trials by having a evidence-informed or data-driven approach to clinical trial design. And then when you're talking about the regulatory environment and commercialization activities, genetic data in the form of genomic landscapes can expedite FDA approval and other regulatory aspects, as well as decrease time to market overall. So genomics holds a great deal of promise in multiple aspects of drug development with specific relevance to rare disease. So let's talk about what is needed to inform drug development in the era of precision therapy. What is required is a comprehensive understanding of genomic biomarkers. And you can break that down into three categories that are undoubtedly familiar to you. The biomarkers being relevant for diagnosis, and what's required there is a clinical grade, industry standard, comprehensive variant interpretation for all genes associated with development of a given disease. An understanding of the functional consequences when that protein or gene product is damaged in some way by those genetic variants or other genetic lesions such as fusion events or copy number change. But what's the functional evidence that indicates that those variants contribute to disease? And do they do so in a uniform way, or is there heterogeneity to those effects? And what's the evidence to discover that information? And then lastly, culminating in understanding how therapies, either known therapies or investigational therapies, how those therapies can be brought to bear to treating the patient. If you're working on an investigational drug, it behooves you to have an awareness of the previous therapies that have been tried and their relative effectiveness. All of that information put together is what is needed for driving precision drug development. But the problem is that manual extraction of this information from the scientific and clinical literature is nearly impossible, as undoubtedly many of you have experienced, and that's where that information is locked down. And where individual data points and studies are available, and readily so, the whole constellation of that information is extremely challenging to bring together and to refine and to annotate and to synthesize manually. And so that's what Genomenon specializes in, and what I'm going to illuminate here when I talk about how we assemble comprehensive genomic landscapes. So another way to describe the challenge, and by threading together some of those slides that I've just discussed, is identifying and interpreting the genes and variants and their attendant therapies, investigational or commercial, and everything in between, that are associated with rare and genetic diseases, including constitutional conditions and hereditary cancers. So identifying and interpreting all of that information allows you to understand what the drivers of disease are, which variants are clinically pathogenic, what functional studies allow you to divine the mechanism of action for your drug or the mechanism of pathogenesis for that disease at the hands of changes in that gene or those genes, what therapies are efficacious, and critically, what's the supporting evidence. That together is the challenge that we're talking about. And when solved, when you've broken this manual research challenge, which is time consuming and error prone, that information can lead to a doubling in your drug development success rate. So Genomenon's approach is to eschew the manual approach, and rather than researchers going out to that information piecemeal, one hypothesis at a time, manually collecting and annotating and synthesizing that information, instead, we have taken an automated approach to having the data come to us to promulgate these comprehensive genomic landscapes, which obviously speeds the efficiency of that process, but also eliminates the errors, whether they're errors of lack of awareness or typographical errors or just plain false negatives. So our computational approach is much more efficient and accurate, leading to nearly 100 times more content coverage and 50 times faster production of this information. And the way that we do that is with the Mastermind Genomic Association database, which is a comprehensive index of genomic evidence annotated for both clinical and functional effect, right down to the variant level, decorated with any information about diseases, phenotypes, therapies, biological phenomena, or any other categorical type of keyword that is necessary to understand your disease in context. Layered on top of the Mastermind Genomic Database are ancillary repositories of information, such as population frequency information and in silico models of pathogenesis, prediction models, which are a core component of many of these clinical standard genetic interpretation guidelines. So Mastermind has mastered the genomic evidence in the decades worth of medical publications. 31 million titles and abstracts are at our command, and nearly 8 million full text and a large number of nearly 2 million supplemental data sets are also exhaustively indexed to identify these terms: genes, variants, diseases, drugs, phenotypes, etc. And specifically with relevance to what we're going to be talking about for rare disease, we have 6.1 million genetic variants that we understand the evidence supporting their disease causation and their role in clinical presentations. Another way to summarize this is in that diagram on the left, which allows me to discuss what a genomic association is. So any one of those entities on the left, genes, variants, diseases, therapies, phenotypes, or categorical key terms, can be interrelated. A gene can be associated with a phenotype. Genetic variants can be associated with diseases. Genetic variants and diseases can be associated with therapies, et cetera, et cetera. Mastermind is a database of all of those associations, and Genomenon is able to surface those associations de novo, or otherwise investigate specific questions that our pharma partners have about any aspect of their disease indication. And the greater number of those associations in the empirical evidence, the stronger that association is, and the more certain you can be in your pursuit of that targeted compound or that candidate biomarker, et cetera. So that's an overall view of what a genomic association is and how it strengthens the evidence to provide confidence in moving forward with your target. What Genomenon does is deliver that high-value genetic insight that comes from a complete awareness of all of those associations. And we do that in two ways. First, obviously, by organizing that evidence, as I spoke to earlier, where most of it comes from the published medical literature, but is supported by any information from those databases that I described. But then beyond just organizing that evidence, Genomenon also identifies those associations. So what you walk away with is that high quality insight from those decades of empirical studies that are necessary to ensure that you you have a highly likely successful drug development strategy. So let's talk specifically about what a comprehensive genomic landscape is. It's a complete data set of all genetic variants for any gene that has been curated, predicated on the evidence in the Mastermind database. And it's curated by humans, as I'll talk to in a slide or two here, to clinical standards, which are ACMG for constitutional disease or AMP for somatic disease. And what we produce in these genomic landscapes are referenced insights for clinical studies, for functional studies, for treatment and trial information, where any clinical or scientific assertion is supported by evidence. And it's summarized in an immediately actionable way, a tiered approach, where we give you a top-line view into which variants are disease-causing or befit the specific questions that are idiosyncratic to your disease or gene of interest. And then the data supporting that ultimate interpretation is categorized and enumerated, and any of the specific assertions are defensible by pointing to the primary source of that evidence, which is to say the individual publications, with an actual quotation to support the inclusion of that study in that categorical. And then critically, this data can be updated quarterly with any of the new findings that appear as literature marches on at the hands of new research. So Genomenon has a unique two-phased approach. As I suggested, we have an automated approach to aggregating, annotating, and assembling all of this information. But once assembled in that way, we follow that work up with an expert level of manual human curation. And we have internal tools that allow us to do that 50 times faster than would be possible if you didn't have Genomenon's full suite of tools at your disposal. So let's, as I said, spend the majority of this webinar talking about some specific case studies in rare and genetic disease to bring to light some of these concepts that I've been talking about and the value of a genomic landscape. I've broken it down to the benefits of a genomic landscape into two buckets. The first is driving R&D activities, and I'm going to speak to using the landscapes to provide insight into mechanism of action, as well as to assess or identify new targets for drug development. So that's on the R&D side. And then on the clinical side, I'm going to speak to specific use case examples that have facilitated improved patient identification, have informed clinical trial design by identifying more candidates who are appropriate for enrollment in the clinical trial. And then lastly, talking about the development of a companion diagnostic to support regulatory submissions. So let's talk about mechanism of action first, which provides these bullet-pointed amenities, one of which is focusing on high-yield candidates and decreasing failure rates. The specific example I'm going to speak to here is Gaucher's and Parkinson's disease, both of which are caused by mutations in the GBA gene. This particular work that we did was tasked with identifying all of the genomic drivers in GBA associated with either or both of these diseases. And the baseline starting point that we began with was a client's awareness of about 3 dozen variants that had been assembled manually over many man-years of scientific literature investigation. So our task in this was to assess the quality of that output. The client that we worked with presumed that what we would provide is ballast for their assertions for each of those 34 variants. And what in fact happened is that there was a subset of those variants for which there was no evidence or contradictory evidence of pathogenicity and disease association. So that was a value that we provided. But then for the rest of those 3 dozen variants, we provided all of the evidence assembled, categorized, enumerated, and assessed that they needed to have confidence that they were on the right track. But in the process of doing those 2 things for those initial variants that the client had awareness of, we uncovered nearly 9-fold additional variants, about a third of which had functional evidence supporting their association with disease, and we did so in a matter of 2 weeks. So if you reflect on the starting point, we have expanded the universe of understanding by first clarifying what exactly they knew before, but opening their eyes to nearly 10 times more information, which provided unique insight into the disease development and then guided some of the downstream development activities up to and including some of the clinical trial strategies that were employed. So this is what I'm going to be showing for most of these examples. This is a comprehensive variant landscape where on the bottom, the x-axis, you can see the protein domain structure, where the linear axis is the different residues of that protein. Then on the y-axis is the number of the variants that were seen at any one of those positions, demonstrated by those different colored dots. So each one of those variants that I'm going to show is a pathogenic variant supported by evidence that may have come from one study or hundreds of studies, but each surpasses the threshold, the clinically acceptable standards for ascribing to that variant pathogenicity. So this is the wheat. This is the good stuff that you need to drive your 2x improvement in drug development success. This is the genomic landscape with all of the evidence predicate. And what you can perhaps appreciate here is that with a complete picture of the comprehensive genomic variant landscape, and only then do patterns emerge in the data, where you can see the peak here for these collections of dots as they aggregate in different domains is a reflection of where the protein is recurrently mutated. And you can imagine to yourself how these variants in their three-dimensional structure conform to a binding pocket or a functional aspect of this protein, which then leads to understanding the mechanism of action if your drug is successfully targeting this mutated protein. So beyond understanding the driver mutations in this gene, we also looked at the data more closely to put a finer point on the clinical ramifications of these different variants. So each row of dots here reflects a different disease, Parkinson's on the bottom and Gaucher's in blue, and then the subtypes of Gaucher's in the various colors above. And what you can see here is, again, with this complete picture, you're starting to see patterns emerge from the data, where the question at hand is, Gaucher's disease is caused by mutations in GBA, but only one subtype has a phenotype related to Parkinson's, a neurophenotype. Why is that when GBA mutations cause Gaucher's disease? Well, what this comprehensive landscape broken down by clinical subtype was able to evince is there's a reason why there's an association with Parkinson's development in Gaucher's type 1, and it is because of the nature of the variants and where they appear along the protein. And that wasn't clear without the genomic landscape work that we did. So the second example I'm going to speak to is in the realm of new target assessment, specifically understanding the molecular basis of disease. And the example here happens to also be in the realm of neurodegenerative disease. This is ALS, one of the causative genes being the TARDBP gene. And the task at hand was to start with an awareness of about 20 variants from ClinVar, which was all the client was aware of, and dig deeper into the decades of empirical literature and uncover more variants that were disease causing. And in fact, we quintupled the number of pathogenic variants that were defensible by evidence, many of which had functional corroborating evidence to ascribe pathogenicity to this protein. So the takeaway from that top line is that ClinVar does not provide the whole picture. And here's a great illustration of that, where on the top in gray you can see those variants in ClinVar, arrayed along the linear axis of the TARD BP protein. And on the bottom in blue are the variants that Mastermind's comprehensive genomic landscape was able to uncover, and again, annotated exhaustively with clinical and functional insight. And clearly what you can see here is that ClinVar is not providing the whole picture. Not even where it does have evidence is it providing the whole picture, but even more fully, there's variants, there's whole classes of variants that lead to disease that ClinVar is not aware of. And so producing this comprehensive genomic landscape can uncover previously unknown vulnerabilities in your target identification approach. The third example I want to speak to is informing patient identification, increasing the diagnostic yield, and critically identifying patients at the point of diagnosis, which is particularly important in the context of rare disease, where the clinicians who are seeing these patients may not be fully aware of the nature of that disease, and the patient may be on a diagnostic odyssey. Similarly, rare disease clinical trials are challenged by the very rarity of the disease itself, and the challenge in collecting enough patients who are accurately diagnosed and likely to respond to the drug, both of which can be facilitated and optimized with a comprehensive genomic landscape. So the example in this vein is Wilson's disease, which is caused by mutations in ATP7B. The starting point would be 250 variants in ClinVar, most of which don't have empirical evidence, most of which have low star ratings, were seen by one lab, were subjectively scored, and are difficult to reproduce. And what our deliverable was able to provide is a 2.5-fold increase in the number of variants that were identified. With all of those variants, ClinVar and additional variants that were only uncovered with Genomenon's approach, all of those variants now have the ballast of evidence, 150 of which had functional evidence, which was informative for ascribing disease causation to these different variants. So in this case, you can go from a relatively low yield patient identification strategy to doubling the number of patients who are identifiable if you have a genetic approach to enrolling your patients in trials or in a companion diagnostic for drug prescription. The second example I want to spend a little bit more time on it was work that we had done with an early client of ours, a continuing client of ours, and it has to do with the comprehensive genomic landscape informing clinical trial design or candidacy at the patient level. Specifically, genomic biomarkers can be used as inclusion criteria, as in this example, or otherwise as exclusion criteria, to make sure that not only do you have enough statistical power to drive interpretability of the study, but you can ensure that you're not diluting the outcome of that trial by including patients who you could have otherwise known a priori would not respond to your drug. So this example is in the realm of monogenic obesity, which has multiple varieties, each caused by mutations in a specific gene or gene set. There's, say, Bardet-Biedl syndrome, Alström syndrome, and then a variety of different gene-specific deletion syndromes. The landscape that was known to the client before we began this work together was causation at the hands of 3 genes, where that manual data interpretation and aggregation process resulted in 12 variants. When we began the work of assembling a comprehensive genomic landscape, we uncovered a great number of additional genes outside of those core 3 that were definitely going to change the way the clinical trial could be conducted, and took the number of variants for inclusion criteria from 12 to nearly 700. So this was a dramatic expansion in this client's ability to enroll patients in their clinical trial, where each of those variants and each of those patients had evidence to support the likelihood of their response during the conduct of that clinical trial. So this client, now a colleague of ours, used Genomenon's Genomic Landscapes to dramatically increase patient enrollment and modified their enrollment strategy so that now it's being conducted on a rolling basis as this new information is coming to light, as new enrollment criteria are being divined. I think the last example that I have speaks to how these comprehensive genomic landscapes can facilitate regulatory submissions by establishing objectivity with genetic evidence and providing evidence predicate for any of the assertions that are made for a companion diagnostic or for understanding things like pharmacogenomics. So this is an example of the RET gene, which is involved when mutated in a hereditary cancer syndrome known as MEN2, in addition to other diseases, but the focus here was on this hereditary cancer syndrome. So not exactly a rare disease, but a genetic disease, and a genetic disease where identifying the information was a challenge. As you can see here in gray, what was known before we began this work was, again, about a 3 variant universe that when we assess the content of Mastermind using our computational approach and our expert curation, we uncovered nearly 4-fold more variants, each with documentary evidence to support their candidacy for inclusion as a companion diagnostic. And another aspect that I'd like to bring out is only with this complete landscape did patterns that weren't otherwise apparent become known. You can see a couple of places where there's no or very little evidence to support the idea that there's a pattern worthy of targeting or worthy of including as an enrollment criteria. And with a comprehensive landscape, that information became readily apparent. And so the takeaway here in the context of regulatory support is that we Genomenon, in producing these genomic landscapes, can help speed the regulatory submission process. And just to give you a specific takeaway of what one of these landscapes looks like, this is a view into Mastermind where we deliver these data. This is for that RET project that I just described, where each row is one of those genetic variants. Each of those variants has a walk-away actionable provisional call based on the assembled evidence that has been expertly reviewed. Those red boxes there are categorizations of the evidence supporting that call with an enumeration of how many studies support that categorization. And then a drop down to showcase all of that evidence at a very granular level that support those assertions that culminate in that overall provisional interpretation. And you'll note a couple of features here. As I just described, the evidence is tiered, so you can dig in as deeply as you need to. You can search through this information and sort it and refine it to empower downstream discovery work, as well as just use the top line information as a walk-away to inform some of those aspects that I described in the previous examples. We can decorate the data in a custom way by disease, by phenotype, by functional consequence, etc. We take in those databases that are necessary to fully inform overall variant interpretation. And then critically, we cite the primary source of the evidence, the peer-reviewed scientific studies, and we encapsulate in this deliverable exactly why that study was informative for any one of those different categories. So this is a very compact and powerful summarization of what the comprehensive genomic landscape looks like, and this deliverable drives those different aspects that I touched on in those specific use cases. So to summarize, the specific benefits of these genomic landscapes can be broken down into optimizing research and discovery activities, divining mechanism of action, or informing new target identification or assessment, or otherwise on the other side of the drug development pipeline, optimizing both clinical or commercial activities by facilitating expanded patient identification, which is particularly important in rare disease, by dramatically refining, informing a clinical trial candidacy for patients, or otherwise changing the whole enrollment strategy altogether, as well as producing very rapidly and comprehensively a companion diagnostic or otherwise regulatory support documentation, all of which can culminate in a twofold increase in the success of a drug program. So the last thing I want to speak to is, some of you may be aware that Genomenon has two sides of its business. One side that you can see on the bottom there is the Mastermind Genomic Search Engine, which is used in clinical genetic testing labs to operationalize the use of this information one variant or one patient at a time. And critically, that genomic search engine, Mastermind, informs both patient diagnosis and treatment, improving the accuracy of diagnosis and reducing the time to treatment. And then the upper arc is what I just described in the way of Genomenon producing these genomic landscapes to inform pharma and biotech for those typical use cases and more that I described. And what's exciting to begin to be talking about is that there's an interconnectivity between these two markets, these two groups that Genomenon serves, the genetic testing labs and the pharma market. And what I'm talking about specifically informs rare disease patient identification, where clinicians and variant scientists using Mastermind, as they see these patients with rare disease, as they see their genetic variants in their workflow, the work that we've done to produce these comprehensive genomic landscapes, as it can be seeded into the Mastermind Genomic Search Engine, will ensure that those patients are no longer missed and that they're efficiently and accurately diagnosed and treated. And so that's work that's ongoing that our attendees today can look forward to in the months and year or two to come. So Kate, hopefully that was audible and intelligible to all.
Yes, I believe so. Yes, thanks, Mark.
I'm glad that we have some time left over for any questions that are on the minds of the attendees.
We do have some questions.
Great.
Can you speak to the approach to cohort analysis that you take to determine the molecular drivers of the diseases?
That's a great question. So just to summarize it here, a cohort is a group of patients who are united by some phenomena. And in this case, I'm presuming it means disease. All of these patients have the same disease. If what we're talking about is each of those patients has had some sort of genomic profiling, the question is, how do we know what gene or variant or sets of variants or sets of genes are driving pathogenicity of that disease, and use that information to inform drug development? So the first approach that we take is to aggregate all of that data and look for recurrence. So in a simple example, say for hairy cell leukemia, if you were to produce a cohort of those patients and sequence at the exome level, say, what you would see consistently across, I think, 99% of those hairy cell patients is a recurrent BRAF V600E mutation. So that happens once in a century. That very rarely happens where you have a singular variant that is highly penetrant across all patients with a given disease. What actually happens more regularly is the same gene is recurrently mutated, or otherwise the same pathway is recurrently mutated, or separately, there's a biological effect that's brought about because of mutations or alterations in multiple different genes and different pathways that converge on that effect. So those are the different ways that you could demonstrate meaning out of a cohort analysis. And Genomenon's approach, after uniting that data and assessing that those relationships that I just talked about, would be orthogonally to take the data in Mastermind and annotate the cohort data in those different categoricals with the pre-existing evidence that allows you then to prioritize which of those aspects, which of those types of ultimate value from the cohort you might see, and then answer every putative hypothesis about this gene or this pathway or this biological phenomena actually contributing to disease in that cohort, all supported by that evidence from the genomic associations from the Mastermind database. So we have a very salient, reproducible approach that's, I hesitate to say it's hypothesis neutral, because it actually answers, asks and answers every single possible hypothesis, and does so with the ballast of the evidence from the Mastermind database. So if the asker of that question would like to talk in more detail about their specific challenge, I'd be happy to go in more depth.
Great, thank you. What are some differences between analysis of constitutional diseases as compared to oncologic diseases when assembling genomic landscapes?
That's a great question. The most obvious difference would be the framework, the interpretation framework, which there's a movement to unite them. I think the two frameworks separately are now talked about in the same breath as ACMG AMP framework, but they do differ in practice clinically. So ACMG is the American College of Medical Geneticists and Genomicists, and that governs interpretation of variants in the constitutional realm, including hereditary cancer. And then on the somatic cancer side, the Association for Molecular Pathologists, or AMP guidelines, dictates how you interpret the variants. And on the ACMG side, the data that is assessed comes in two flavors: internal to the case or the variant, and external to the nature of that case and the variant. Which is where Genomenon brings in these population frequency databases, the in silico predictive models of pathogenicity, and all of that evidence from the publications. But we take all of that information together and promulgate a provisional call for each of the variants that we've exhaustively identified by reviewing the data that's indexed in Mastermind. So that's ACMG. AMP, the approach is a little bit more simple, but you have tiers of, is this in the standard acceptable guidelines like NCCN or FDA? Otherwise, you look at whether it's in routine clinical practice that this variant is diagnostic and associated with a therapy, or if you're not able to have that information because it doesn't exist, there's the assessment of the information for other clinical studies or functional corroboration that this variant is disease causing, and that information leads to a tier 3 or tier 4. So there's just a different framework approach to assessing those two different types of diseases, but Genomenon is expert in both of those and can deliver a comprehensive genomic landscape for each. So that's sort of operationally. From a content perspective, I'll say that in constitutional disease, there's usually a more idiosyncratic disease causation in surrounding clinical parameters that need to be assessed, whereas in oncology, there's more of a stereotypia. It's usually much more uniform what you're looking for, even though the cancer may be different. There's less of an idiosyncratic or custom approach to assembling this data, though we can and have, for those somatic cancers, produced more custom annotations to the comprehensive genomic landscape. And the last thing I'll say about how somatic cancer differs is that obviously cancer is economical and reuses mechanisms. So when you're putting together a data set with a specific disease indication, it's often beneficial to take a step back and look at other diseases that also result at the hands of the gene that you're specifically targeting, even if it's not for your primary indication. And as I said, given that it's a comprehensive genomic landscape, we include all of those additional diseases that are peripheral to but still informative for your core disease indication.
Great, thank you, Mark.
Sure.
Another question. How are variants without empirical evidence incorporated into genomic landscapes?
That's a great question. If there's no empirical evidence, they're still included by virtue of the fact that they have been identified in large population studies, say. So if they're benign, or you could consider that a GWAS variant or a variant identified in GWAS lacks proper empirical studies or evidence, but is nevertheless a variant that has information associated with it. Both of those types of variants are included. We are also able to go out to locus-specific databases where users or clinicians have submitted variants that they've identified for which there's no proper peer-reviewed study. So our approach in assembling a comprehensive genomic landscape is to bring in and aggregate all of those lines of evidence, and then to, as I mentioned throughout, decorate with the empirical studies to strengthen those associations and provide ballast for and confidence in your decisions moving forward. Another note to make about variants that you identify in clinical workflow for which there's no specific evidence about that self-same variant, there's benefit in assembling these comprehensive landscapes because related variants related to the variant for which there's no empirical evidence, if those variants that are related to that variant do in fact have empirical evidence, that evidence can support a promotion of this variant from uncertain significance to likely pathogenicity. And that kind of information can only be afforded by putting together a comprehensive genomic landscape where the sheer burden of and comprehensiveness of that evidence in the comprehensive genomic landscape can then promote those variants for which there's otherwise no empirical evidence. So that's the way that I would answer that question in those two different approaches.
So then how can those landscapes help to better identify rare disease patients?
That's a great question. So and that's actually I put in that last slide about Genomenon's core business and how it bifurcates. I put that in the last minute, and this question allows me to go into a little bit more detail. But at the hands of a clinician, whether they be a pathologist or a geneticist or not even a clinician, but a variant scientist, somebody looking at the genetics of a patient, there's a need to have the most comprehensive information to maximize the diagnostic opportunity. If you don't have a comprehensive landscape, you run the risk of this patient being missed, even though the sequencing data contains an informative variant. If you don't have a complete aggregation of all of this information, that self-same variant or the variants that are related that have evidence that support that variant would otherwise be missed by the clinician or the variant scientist looking at that information. And so all of that evidence that we assemble comes along with the appearance of that variant when searched for in Mastermind's genomic search engine or otherwise through our partnerships with tertiary analytic software, where that information gets threaded through and fed forward and can inform the prioritization mechanism in those software packages to say, this is a variant that you can't miss, and here's the evidence supporting why, and it's associated with a rare disease, which, by the way, has an efficacious treatment, or otherwise has clinical trials that the patient can be enrolled in. And so that's where we have this exponentiation of the value that we can bring to the genomics market writ large, is in producing these comprehensive genomic landscapes to drive drug discovery and clinical trial design in pharma, bringing that comprehensive knowledge into the Mastermind search engine and surfacing it to users, either direct users of Mastermind or through our partnerships with these different software providers, will then ensure that those rare disease patients aren't missed anymore.
Okay, great. Another question. I'm going to make sure I get this right. Okay. Can the Genomenon data be used with human microbiome information?
That's an interesting question. Used with is pretty loaded, and so it's pretty wide open. So I'll answer it in a couple of different ways, but sticking to that wide open territory. Referring back to the genomic association data that I mentioned before, Genomenon's genomic landscapes needn't be predicated on individual human variation. So if the question in the context of microbiome and infectious disease, say, was more about what are the species of infectious agent that are associated with different diseases or different phenotypes, that is an association that we can put together and have it be comprehensive. So our mastery of the empirical evidence allows us to ask and answer those types of questions. More specifically, if there's a question about the way that human genetics informs or is informed by different findings in metagenomics or the microbiome, when we produce the comprehensive genomic landscape, each of those variants that comes to the fore as being associated with this disease or being found in this gene, if the question is more specifically, which of those variants have any empirical evidence to indicate a relationship between themselves and metagenomics or GI flora, say, if that's the specific custom annotation that's being asked for, that is definitely something that can be part of a genomic landscape deliverable. I'll encourage whoever asked that question to arrange a follow-up with me. Actually, I can be reached at this email. So if that answer was too vague, I welcome the opportunity to dig in deeper and talk specifically about your use case and to walk you through some of the examples that I was just lightly touching on in my answer.
Great, terrific. All right, well, thank you, Mark. Thank you for your time and all this information you've shared with everyone. And thank you, everyone, for joining us today. We're going to be sending a follow-up email with a link to this recording and the Q&A portion of today's event. As Mark mentioned, if you'd like to connect with him, you can email him at this email here. It's mark.kiel@genomenon.com. And make sure you're following us on Twitter and LinkedIn at Genomenon. And thank you again. Enjoy the rest of your day, everyone.
Thanks all.